I'm Sue Branford. I work at the Centre for Cancer Biology, SA Pathology in Adelaide, Australia, and I'll be talking about current and future techniques with a particular focus on drug resistance testing. CML is a genetically uniform disease, and the BCR-ABL fusion is at the heart of it. Patients treated with tyrosine kinase inhibitor therapy can have a near normal survival. However, not all patients respond optimally. A proportion of patients fail therapy due to drug resistance, and some patients progress to blast crisis, where treatment options are limited. And BCR-ABL transcript kinetics can identify patients with pending resistance. Most patients have a rapid decline, as shown here in this example of BCR-ABL transcripts measured over time using an international reporting scale. 70 to 80% of patients have this optimal response, and these patients are essentially protected from disease progression. But some patients have a slower decline, and they're at risk of treatment failure, which is signaled by a rise in BCR-ABL transcripts and acquired resistance. And some patients have no response at all, which is primary resistance. So as these patterns of molecular response may signal treatment failure, where a change of therapy is mandated. The major known mechanism of acquired resistance is mutation within the BCR-ABL kinase domain at the drug binding site. And shown here is the BCR-ABL kinase domain crystal structure in complex with imatinib in green. The red circle is a representation of a mutation, and these can occur at amino acid that makes direct contact with imatinib. But imatinib binds with a very tight fit, and many mutations throughout the kinase domain can interfere with imatinib binding by altering the conformation of the protein. And here is a linear representation of the kinase domain and the location of various mutations. Among the most common are located within the phosphate binding loop, the P loop, and some of these have been shown to be more transforming than wild-type BCR-ABL. T315I is known as the gatekeeper mutation because it forms a critical hydrogen bond with imatinib, and the second-generation TKIs with all of these, and it causes resistance. So kinase domain mutations are detected in about 50% of patients with acquired resistance, but they're not the cause of primary resistance. And drug resistance has driven the development of more potent BCR-ABL inhibitors such as nilotinib, dasatinib, bosutinib. But specific BCR-ABL mutations can cause resistance to each of these inhibitors, including T315I. And the US-based National Comprehensive Cancer Network regularly publish updated guidelines for CML, which includes a table of the contraindicated mutations for each of these inhibitors. For example, F317L is sensitive to nilotinib. But what if a patient has an F317L that is dasatinib and bosutinib resistant, plus an E255K nilotinib resistant mutation? Well, ponatinib is available, where T315I is the only single molecule that can cause resistance. But the resistance profile of compound mutations, that is mutations that occur on the same molecule, is unpredictable, and certain compound mutations confer ponatinib resistance. And in these cases, allogeneic transplant may be indicated or experimental therapy. For example, the new inhibitor asciminib that specifically targets the myristoyl pocket of ABL and is currently in clinical trial could be used, but there is a specific new spectrum of BCR-ABL resistant mutations that have been identified in patients treated with asciminib after failure of other TKI therapy. So characterizing the type of kinase domain mutation is essential for treatment decisions, and BCR-ABL kinase domain mutation analysis remains a critical component of patient management. So what about the recommendations for when to test for mutations? Well, these are at failure to reach response milestones, any sign of loss of response defined as hematologic or cytogenetic relapse, a significant rise in BCR-ABL transcript, and disease progression to accelerated or blast phase. And I'd like to emphasize that there is no indication to perform mutation analysis at chronic phase diagnosis. Furthermore, the recommendation is to always collect a sample for mutation analysis before TKI therapy is ceased, because unmutated BCR-ABL can rapidly and preferentially proliferate in the absence of BCR-ABL inhibition, and this can lead to a false negative mutation result. Capillary Sanger sequencing has been the mainstay of detecting kinase domain mutations over many years, where the sensitivity is 10 to 20%, and the kinase domain of the BCR-ABL allele can be specifically amplified using RT-PCR. Shown here is the linear representation of the normal ABL allele and the BCR-ABL allele, and each leukemic cell normally has one copy of each, and the ABL kinase domain is common to both. So PCR primers are designed to hybridize within a BCR exon and downstream of the kinase domain. And this strategy excludes amplification of the normal ABL allele. And by excluding the wild type ABL allele, BCR-ABL kinase domain mutations are detectable in samples with very low BCR-ABL values of approximately 0.1% on the international scale and above. The amplified product is about 1,500 bases, and this can be visualized using gel electrophoresis. And the kinase domain is sequenced using PCR primers that are located just outside of the kinase domain. And Sanger sequencing can track the clonal expansion of mutants over time. Shown here are sequencing chromatograms demonstrating the rise of a mutation over time, and this mutation is Y253F that is located within the P loop. Now, some mutations can emerge and become the predominant clone very rapidly and cause overt relapse, as occurred with this mutant. So by detecting mutations early, this is very important because a rapid switch of therapy can be implemented to reduce the risk of resistance. And we know that this mutation is resistant to imatinib, but it may be sensitive to second-generation TKIs, and a rapid switch could prevent the clonal expansion of the resistant mutant. But more sensitive methods enable the early detection of kinase domain mutations, and ideally when they are still at a low level before clonal expansion. And the first method to demonstrate the importance of detecting low-level mutations was high-throughput multiplex mutation genotyping using a chip-based mass spectrometry. This is a primer extension assay that has a sensitivity of 0.05 to 0.5% mutant. So it's greater than 2 logs more sensitive than Sanger sequencing, but the method is only semi-quantitative. And unlike Sanger sequencing that allows an unbiased examination of the kinase domain, only selected mutants are detectable using mass spectrometry. But the assay can be designed for the most frequent mutations detectable in patients, and we developed an assay that detected 91% of all mutations that cause resistance to imatinib, nilotinib, dasatinib, and bosutinib. And the assay redesign is required with the emergence of mutations that uniquely cause resistance to newer BCR-ABL inhibitors. Mutations below the detection limit of Sanger sequencing were found in 29% of patients with imatinib resistance using mass spectrometry, who were subsequently treated with nilotinib or dasatinib. And low-level mutations detected at imatinib resistance invariably expanded with second-line nilotinib or dasatinib treatment if they conferred resistance, such as the T315I or the F317L mutation, which is dasatinib resistant, or the Y253H, which is nilotinib resistance. and the mutations expanded rapidly and were detected by Sanger sequencing at a median of 3 months. So this work demonstrated that low-level mutations are clinically relevant, but mass spectrometry is not widely used. We also demonstrated using this method that the relevance of multiple low-level mutations is significant. So patients with more than one low-level BCR-ABL mutation had a poor prognosis, and this was irrespective of the sensitivity profile of mutants. Shown here are 2 patients that we monitored at our institution. Both had imatinib resistance related to a predominant G250E mutation, and were treated with nilotinib because this mutation is known to be sensitive to nilotinib. The patient on the left had no low-level mutations and had a good response, as shown in the plot of BCR-ABL transcript values, whereas the patient on the right had 7 additional low-level mutations and a poor response, and a new mutation, the T315I, also emerged. So having multiple low-level mutations may be a marker of genomic instability, and their detection is clinically relevant. And again, this work has demonstrated that low-level mutations are clinically relevant, but again, mass spectrometry is not widely available. So more recently, next-generation sequencing techniques have been adopted for BCR-ABL kinase domain mutation analysis. And NGS provides enhanced sensitivity for BCR-ABL mutation detection with a limit of detection of about 3%. However, distinguishing low-level variants from background artifacts remains one of the main challenges for next-generation sequencing, and careful optimization is required. NGS detected early appearance of clinically relevant mutations that would not otherwise be detected by Sanger sequencing, and confirmed the prior clinical findings using mass spectrometry. So NGS will allow early clinical intervention and improve clinical outcome. And NGS uses the same initial strategy as Sanger sequencing to isolate the BCR-ABL kinase domain, and 150 or 250 base pair amplicons are sequenced. And here is an example of next-generation sequencing data from our work, where sequencing reads were visualized using the integrative genomics viewer. The T-base indicates a missense variant. The patient had treatment failure and switched to dasatinib. At the time of switch, there was no mutation detected using Sanger sequencing, but NGS retrospectively revealed a T315I mutation at 3% that rapidly expanded to 23% after 4 months of dasatinib. So this is an example of an inappropriate TKI that was used at treatment switch. And a recent study prospectively assessed patients with non-optimal response using NGS, which detected low-level BCR-ABL mutations in patients at the time of a warning response, and these expanded over time. So again, early detection of low-level mutations using next-generation sequencing allows rapid intervention and selection of the most appropriate TKI. Importantly, viability studies of NGS-based mutation analysis was performed across collaborating labs, which confirmed the accuracy and reproducibility of the assay. And the European Leukaemia Net 2020 recommendations for treating CML suggest the greater sensitivity of NGS enables the early detection of clinically relevant BCR-ABL resistant mutations and is the recommended technology to detect BCR-ABL mutations in patients not responding adequately to TKI. But it is important to appreciate that all sequencing techniques can be technically challenging. NGS for BCR-ABL mutation analysis will not be available in all labs, and Sanger sequencing will continue to be widely used, particularly if results can't be reported in a clinically actionable timeframe using NGS. So accurate mutation analysis requires optimization of all procedures. Accuracy for any method will vary depending on a number of factors, including the use of an appropriate PCR enzyme and the amplification efficiency of the PCR. And a critical factor is the quality of the RNA, because poor quality RNA can generate false positive and false negative results. And it's also essential to establish a threshold variant allele frequency, below which samples are rejected for mutation analysis due to the risk of a false positive result. But over the last few years, evidence has accumulated that mutation of cancer-related genes other than BCR-ABL is associated with treatment failure in CML, including primary resistance. And a review of the literature on behalf of the ICMLF Genomics Alliance found 15 studies that reported mutations in blood cancer-related genes in patients at diagnosis. And in our study, we performed whole exome sequencing, RNA sequencing, and copy number variation for 46 patients at diagnosis who were highly selected based on either having a good or a poor outcome, and found a significantly higher frequency of cancer gene mutations in the patients with a poor outcome. So NGS analysis at diagnosis for cancer gene variants is exploratory at this stage because the clinical relevance of these variants has not been established. And there are many questions that remain to be answered, including, do some mutations confer a poorer prognosis? Do multiple mutations have greater impact than a single mutation? Can the poor risk be overcome with more potent drugs? And will mutations at diagnosis impact the chance of treatment-free remission? Now, these are essentially the same questions that were addressed for BCR-ABL kinase domain mutations that required many years and many studies to resolve. So to answer these questions, an International CML Genomics Alliance was formed in 2018 with the aim to harness the wealth of genomic information that is generated in local CML research projects by building a platform to facilitate data assimilation and sample exchanges for improved statistical power. And this was facilitated by the ICMLF and supported by the CML Patient Advocates Network. We've recently partnered with the European-based Harmony Big Data project to achieve our aims. This is a central repository of anonymous data of patients with blood cancer, where access to data is secure and for approved projects only. So we encourage anyone to participate in this project and share their clinical and genomic data. So we aim to identify clinically relevant prognostic subgroups for enhanced risk stratification at diagnosis and discover biomarkers that could predict treatment response and progression and guide drug use and development, and provide data for evidence-based expert international clinical practice guidelines for genomic monitoring in CML. Now, interestingly, the NCCN CML guidelines already include myeloid mutation panel testing in certain circumstances. And the guidelines state to consider myeloid mutation panel for patients with accelerated or blast phase to identify BCR-ABL independent resistant mutations in patients with no BCR-ABL kinase domain mutations. However, at this stage, there is no guidance for treatment intervention if a mutation is detected, and a myeloid mutation panel may not be sufficient for patients with lymphoid blast crisis, where deletions in genes such as IKZF1 are the major mechanism of resistance. And we found that multiple mutations were detected in blast crisis. Again, we performed next generation sequencing, this time for 39 patients in blast crisis, and identified multiple mutation types. Most frequently, frameshift, missense, nonsense, and splice site mutants in 72%, cancer gene fusions in 31%, and cancer gene deletions in 36%. And by combining the different mutation types, we found that 100% of patients in Blast Crisis had one or more mutated blood cancer-related gene, and 58% of patients also had BCR-ABL mutations, and every one of these had additional mutations in cancer-related genes. So BCR-ABL dependent and independent resistance mechanisms frequently co-occur. So to summarize, BCR-ABL kinase domain mutation analysis remains an integral component of patient management. The gold standard method has been Sanger sequencing and will likely continue to be used by many labs in the foreseeable future. Low-level mutations are clinically relevant, and the ELN now recommend more sensitive NGS analysis in patients not responding adequately to TKI. And NGS can also identify additional mutations in cancer-related genes, but again, their clinical relevance awaits validation. Thank you.